Welcome everybody here in 124 and online. We're very pleased to have a very distinguished speaker today. We thank the Center for Integrative Environmental Health Sciences for agreeing to switch places with us as we have a faculty candidate coming in next week when their seminar usually is. So. But today's speaker is more than eminently qualified to provide a seminar relevant to environmental health sciences. Sanjay Srivastava is a distinguished university scholar and professor of medicine with secondary appointment in pharmacology and toxicology at the University of Louisville. He's also director of the University of Louisville Superfund Research Center, which many of us participate. He obtained his bachelor's degree in chemistry and his Ph.D. in chemistry from master's in chemistry and Ph.D. all from Lucknow University in India. His Ph.D. focused on oxidative stress and platelet functions. He did his postdoctoral training in protein chemistry at the University of Texas Medical Branch at Galveston. In vascular biology and toxicology, and he has started his career here at the University of Louisville in 1999, and he has risen to the rank of professor in 2012. He's extremely productive. Since 2002, he's published over 140 research articles. He's also chaired or been a member of more than 50 NIH study sections. That's brutal. And on the editorial board of Circulation Research, one of the high impact journals in cardiovascular medicine. So the focus of his group has been to elucidate the cellular and molecular mechanisms by which endogenous and environmental carbonyl compounds affect endothelial function, vascular inflammation, and atherogenesis. So Sanjay is one of our leaders, eminent leaders here at the University of Louisville, and he's one of the good guys who is always willing to collaborate and to work with others. So it's an honor to introduce him. He's going to present his research on carbonyl stress in cardiometabolic disease. Sanjay.
Thank you, Dr. Hein, for the invitation to share some of our research work with you guys. And this seminar is a little different than the usual seminars I give because some of the younger folks talked to me about when I was giving a talk actually at Yale a couple of months back, and they wanted to see like what your career track was and what are the challenges you face as you develop your career and what got you to where you are. So I have included some of those elements. Let's see like from where I started and how the research whole program developed. So before I give my presentation, I want to
There's coming.
Oh, there it is. Okay. I want to dedicate today's talk to my PhD mentor, Dr. P.K. Seth, who passed away last month. Dr. Seth was a very eminent scientist, neurotoxicologist in India, and he was even a better person. He spent a lot of time at University of Chicago and NIH, and then went back to India and established the world-renowned neurotoxicology program at the Institute of Toxicology in India. So I dedicate my talk to him. Now, as you know that the focus of my research group has been to study the cardiometabolic disease. And the reason is that diabetes is a problem which is exponentially growing worldwide. Between China, India, and US, we have more than 200 million people who are diabetic. And similar to diabetes, heart disease is also a major problem, which accounts for a large number of death, and perhaps it's the leading cause of death worldwide. The leading cause among the heart disease is cardiovascular disease with coronary artery disease and stroke. Stroke is more prevalent in China. And we don't know why this like a stroke is more prevalent in China, whereas in India and in the US and rest of the world, coronary artery disease is the leading cause of death. But the mechanism of pathology is more or less similar. So if we talk about cardiovascular disease as the leading cause of death, atherosclerosis is the underlying cause of most of the heart disease. Coronary artery disease and stroke account for almost 70% of the total disease and death. And this atherosclerosis, especially in the coronary artery, starts with the endothelial activation, and then it ends up recruiting lipids and inflammatory cells, and you have narrowing of the arteries, and the plaque eventually ruptures, which leads to heart attack. And if the same process happens in the carotid artery, you get stroke. So the focus of my research group is try to understand the molecular mechanisms of atherosclerosis and how are those factors modulated by environmental exposures. So there are several risk factors for the cardiometabolic disease. That includes nutrition, smoking, pollution, physical activity or lack of physical activity, inactivity, circadian regulation, and change in environmental conditions. So the studies, the areas listed on the left side, nutrition, smoking, and pollution, they are essentially regulated by this group of chemicals, what we call carbonyls. And these carbonyls essentially are aldehydes or ketones. So you have carbon with a double bond with oxygen, or you have the aldehyde group. These carbonyls are generated by endogenous ways. Oxidative stress generates a lot of carbonyl compound. Glucose by itself is an aldehyde. It is a carbonyl compound we get from dietary sources. Xenobiotic metabolism, especially some of the drugs and environmental chemicals, generates a lot of aldehydes. And molecules like acrolein are generated by mitoperoxidase-driven reactions from the neutrophils. And then from the environment, we get from combustion, especially from fossil fuel, you get a lot of crotonaldehyde and acrolein. Then you get tobacco products, which are very rich in carbonyl compounds. And a lot of industrial chemicals, either by themselves are carbonyls or they are metabolized to carbonyls. So that's the reason we are focusing on the carbonyl toxicity. And these are some of the examples. So glucose, glyoxyl, methyl glyoxin, and glucosone. These make advanced glycation end product, and that causes a lot of secondary diabetic complications. Then the lipid peroxidation derived aldehydes, 4-hydroxynonenal metaldialdehyde, and acrolein in a smaller quantity, but is generated more during inflammation. And then environmental chemicals like acrolein and crotonaldehyde I talked about. And then molecules like butadiene and benzene, they are not carbonyls by themselves, but they are bioactivated and metabolized to generate carbonyls, and their toxicity in part is mediated by carbonyls. So we asked three fundamental questions. Do carbonyl induce cardiometabolic disease? If so, what are the underlying mechanisms? And then what are the strategies to mitigate the toxicity of these carbonyl compounds? So I break my talk in three parts: my graduate studies, how I started doing this, and then where I studied oxidative stress, essentially how that contributes to platelet activation, and more in pathological conditions. And then postdoctoral studies where I looked at the aldehyde metabolism, and then independent program, and I established over here how the aldehyde generated by oxidative lipids or present in the tobacco products or in the environment contribute to the exacerbation of atherogenesis and cardiovascular disease. So when I started as a graduate student in around 1989-90, the focus of my work was to study how oxidative stress affects platelet function and how those are regulated by some of the environmental factors that I was studying at that time was metals. But unfortunately, at the physiological concentration, we did not find much changes in the platelet functions with the metals. But what we found was the aldehyde generated by lipid peroxidation, which were catalyzed by metals, they led to cross-linking of the proteins. And this protein cross-linking compromised a lot of membrane functions, including membrane polarization, membrane fluidity, which led to increase in intracellular calcium level. And then this was with phosphatidyl kinase different reactions. And inositol was a major part of that. And then the increase in intracellular calcium contributed to platelet activation, especially platelet aggregation. So a couple of studies we did. One study where we looked the effect of diabetic and non-diabetic stroke on these parameters, and we found that indeed platelet activation occurs through this pathway. And if you correct for diabetes, this study showing on the right panel, upper right panel, that if you correct for diabetes, then several of these effects are reversible. We also looked at the effect of oxidative stress and platelet function in restenosis, but we found there was no increase in oxidative stress during restenosis, or there was no increase in platelet activation in the process. So when I moved to Galveston in 1994, my mentors group was studying the role of polyol pathway enzyme aldose reductase.
Yeah. Okay.
Is it okay? Okay. So they were studying this the effect of polyol pathway enzyme aldose reductase on secondary diabetic complication, and there were studies going on which show worldwide, actually at that time, that inhibition of aldose reductase was beneficial for a lot of vascular complications, but there were indirect toxicity of those aldose reductase inhibitors, and those inhibitors were discontinued, except for Japan. I think they are banned everywhere else. So if you look at the aldose reductase, glucose was is supposed to be a substrate, but under euglycemic conditions, glucose metabolism glucose contributes to only 3% of the sorbitol formation by aldose reductase, whereas in diabetic conditions, it increases to 30%. So and the other thing what happened around the time that I joined the group, the structure of this protein was solved in 1993. So when the structure of the protein was solved, they found that the active site of the protein was very hydrophobic and not suited for the molecules like glucose. So maybe glucose was an incidental substrate in the especially in the glycemic conditions. So because of my background in studying lipid peroxidation products, so why don't we look at the aldehyde generated by lipid peroxidation and see how they are metabolized or reduced by aldose reductase. So then we did a series of experiments doing the structure activity relationship. And what we found that the short chain aldehydes were not good substrate for the protein. But when we got a 6 carbon or longer chain length, then they interacted with the hydrophobic residues at the active site, and they were catalyzed very nicely. So for example, the Km decreased from 5,000 micromolar to 10 nanomolar as we went to 6 or 9 carbon. Another thing what we found was that some of the molecules which are unsaturated in nature, they can bind with cellular nucleophiles like cysteine and of the glutathione. And the glutathione conjugate was a very good substrate for the protein. It dropped the Km from 5 millimolar for the 3-carbon compound to 13 micromolar. So this led to a new discovery that the protein has a glutathione binding site, which my mentor's lab later on went and crystallized the protein and showed that. So another thing, what we were doing when we were doing these assays, we found that we were having a lot of trouble doing these assays because the enzyme would sometimes get activated or inactivated. So then we did a systematic analysis what was going on, and what we figured out was the sequence of addition of the substrate was very important. If you add the substrate before adding the co-substrate NADPH, that actually modifies the protein. So we did the mass spec of the protein treated with acrolein or HNE, and we found these molecules bound at the active site of the protein. And what was interesting was that the short chain molecules were activating the protein, like here with acrolein, you can see a 7-fold activation. And as you increase the chain length, it starts to inactivate the protein. So this really told us that several of these molecules are involved in the post-translational regulation of the protein. So complementary to these studies, we started looking at how nitric oxide and other molecules regulate the activity of this protein. So we treated this homogeneous aldose reductase enzyme with glutathione NO, GSNO, and a nitric oxide donor. And the idea was that NO released from the GSNO will activate the enzyme like short molecular weight compound. But what we were seeing is that there was a significant decrease in the enzyme activity. So then we did the mass spec. We were thinking that, okay, maybe this nitric oxide is bound to the active site, and that is contributed to that. But what we figured out actually that it was not nitric oxide which was really tightly bound to the active site, but it was the glutathione molecule which bound to the active site. So that led to the new area of protein glutathionation, and there have been hundreds of studies since then. But this actually was an accidental finding which opened up the new field of protein glutathionation. So when I moved to Louisville, we pursued these studies further because I had mass spec in-house and it made life much easier. And what we found actually was that our original hypothesis was right. Incubation of the protein with other anode donors, which generated nitric oxide, actually activated the enzyme. As we used 2 different molecules, SNAP and glycosnap, to release nitric oxide. So it led to the methodology mass spec based assays to look for the protein nitrosation and protein nitrosylation. I'm not pursuing these studies further, but several of my collaborators have picked up on these. And this is, again, they're looking at the cellular level now. So this field has really grown leaps and bounds. So when I moved to Louisville, then I started collaborating from some of the collaborators over here, trying to see how this aldehyde metabolism is associated with cardiovascular disease, especially in the disease model. So this is the study we did in collaboration with Dr. Boley's group. And essentially what we found that inhibition of aldose reductase prevented the ischemic preconditioning, and that was dependent on the generation of lipid peroxidation derived aldehyde. Similarly, we did a study with Suman Prabhu in the heart failure model, where we found that the heme oxidase 1 overexpression prevents oxidative stress in the heart and that really prevents the heart failure. And this is the study that I did in my lab, where we developed the diabetic stenosis model and look at the smooth muscle cell proliferation, and we found that inhibition of aldose reductase prevented the neointima formation, both at 10 weeks after the injury and 21 days after the injury. However, this restenosis part was independent of increase in lipid oxidation. So it turned out that it was PKC mediated processes which led to NF-kappa B activation, and that led to the proliferation of smooth muscle cells. And when we gave aldose reductase inhibitor, that inhibited those processes. So because we were working with some of these carbonyl compounds, especially 4-hydroxynonenal as a model compound, so we synthesized the radiolabeled compound, tried to understand the actual metabolism, especially in the cardiovascular tissues of these carbonyl compounds. And I'm not going to go into details, but what we found was in the cardiovascular tissues, the reduction by aldose reductase is a relatively minor pathway. Oxidation is a prominent pathway. That's like 20 to 40% of the aldehyde is converted through this pathway. And bulk of the protein of the aldehydes is conjugated with the glutathione. And then glutathione conjugate can be either excreted out by itself, or it is reduced by aldose reductase. And this is a major pathway for the detoxification of unsaturated aldehyde. And these molecules are involved in redox signaling and inflammation. So based on this data, I wrote my first RO, like 2 R01 grants in 2002, and both of them got triaged. And the reason was they liked the concept, but they said, like, so what? You metabolize, whether show that under pathological condition is doing something to the disease process. So then I went to, and by the way, these studies were done in major part by Max Spike, who then, since then, who was a student in the lab at that time, and since then he has moved on to Harvard, where he's associate professor now, having his very good program in looking the regulation of inflammation. So anyway, so when I was told by the NIH study section that I need to develop some pathological model to demonstrate that they have physiological significance, I reached out to Helen Dougherty at University of Kentucky, and with his help, established the model of atherosclerosis in my lab. And what we found there, and incidentally, once we provided the preliminary data, both the grants were funded with like 6 or 7 percentile. So, so what we did here was made the mouse on the ApoE background, these aldose reductase knockout mice with ApoE knockout mice. And the idea was that if you have deficiencies in aldose reductase, it is going to increase the aldehydes in the lesion and in circulation, and that would exacerbate atherosclerosis. Indeed, that's what happened whether you use the pharmacological inhibitor or genetically delete aldose reductase. There is increase in lesion formation. Then the next series of experiment we did is like, okay, if you give aldehyde by themselves, what happens? So exposure to aldehyde like acrolein exacerbated the atherosclerotic lesion formation. And then if you look at the carnosine, one of the molecules which quench these aldehydes, then you could decrease these lesion formation and by decreasing the aldehyde content in the lesions. So essentially, three independent experiments shows exposure to aldehyde increases lesion formation, inhibition of aldehyde metabolism increases atherogenesis, and if you quench the aldehyde, then you decrease these atherosclerosis by decreasing the lesional aldehyde content. So this carnosine study was done by Oleg Barsky, who since then has moved to NIH. So the next question was, okay, so aldose reductase under euglycemic conditions, we can understand that it is contributing to aldehyde metabolism and is inhibiting that is bad. But what happens under diabetic conditions? Because there were a lot of studies which were showing that inhibition of aldose reductase was a good thing. But what we found that even in the diabetic conditions, there were exacerbation of atherosclerosis. Actually, it was more pronounced. And then in the lesions, we see the cross-linking with a lot of age formation, advanced glycation and product, which were, like as I said before, glucose, methyl glyoxyl, these molecules cross-link that. And then if you have accumulation of ages that contribute to secondary diabetic complication. So we found that inhibition of aldose reductase was bad under diabetic conditions, especially under the chronic conditions. They may have beneficial effects short term, but long term, they perturb the aldehyde metabolism and it is not good for you. So since we saw that there was increased accumulation of aldehyde in the lesions, we did a mass spec trying to figure out whether these protein modified by aldehyde are just the footprint of the injury or they are involved in the disease process. And with Mike Merchant's help, we could show that if you treat the endothelial cells with aldehydes, then you modified a lot of proteins. And our hypothesis initially was that there will be a lot of mitochondrial protein and some of the cytosolic protein will be modified because those are the sources of free radical generation. But what we found in addition to those proteins, a lot of proteins which were modified were ER resident, endoplasmic reticulum resident, or associated protein. And this study was initiated by Brad Hill, who is now professor of medicine over here. And subsequently, we looked at the protein modification of what is the role of ER stress in causing endothelial activation and atherogenesis. And Petra, who is here in the audience, led the studies with acrolein, and Elena, another postdoc in the lab at that time, she looked at the contribution of 4-hydroxynonenal in inducing ER stress, and then that led to the unfolded protein response. So here you see the three canonical pathways of unfolded protein response, and all of these pathways were activated by the exposure to these aldehydes. And then we did a series of in vitro studies to tease out the molecular mechanism. And finally, what we did was expose the rats to some of these chemical chaperone which prevent protein folding, and did the intravital microscopy to look at the rolling and adhesion of leukocytes. And we found that if you prevent protein folding by giving this chemical chaperone, then you inhibit ER stress, and then you inhibit the endothelial activation by H&E. So these studies were, these physiological studies and cellular studies were led by Israel Sutu, who is with us for a long period of time. So while we were doing all these studies directly looking at the aldehyde molecules, another question was, other molecules which cause oxidative stress, do they follow the similar pattern? So for that sake, we studied arsenic in collaboration with Chris Tate. And the reason was twofold, that arsenic causes ER stress, that was known, and arsenic causes oxidative stress. So we saw like, well, are these processes involved in arsenic-induced atherogenesis? So we established the model and we found that arsenic indeed exacerbated atherogenesis. It increased the accumulation of these aldehyde modified proteins in the lesions, and there was also free aldehyde in circulation. And if we give again the protein, the chemical chaperone of protein folding, you can prevent the arsenic-induced exacerbation of atherogenesis, suggesting that ER stress could be involved in this process. And when we teased out the mechanism, unlike the aldehyde per se, we found that the PERK pathway was not involved, but IRE1 and ATF6 pathways were involved in increasing inflammation and apoptosis, and which led to atherogenesis. So when we were doing that, we published these studies with the ER stress. Then this was noticed with one of my colleague at one of the NIH study section, Hong Chen, and she was interested she's working on this protein called epsin and atherogenic effects of epsin. So she was interested how this epsin interacts with IP3R1 receptor, and our hypothesis was these processes are mediated by ER stress. So then we started collaborating, and then we were fortunate to get three multi-PI NIH R01 grant funded, which are still active. And we established a collaboration where we are looking at the role of epsin in ER stress, and how now she has synthesized some small molecular compounds, looking how exposure to those compounds mitigate the cholesterol uptake by the macrophages, and how that affects dyslipidemia, and are these processes mediated by ER stress. So while these studies were ongoing, we were approached, actually, Arnie was approached by American Heart Association in 2013, I believe, 2012, 2013. So at that time, FDA came up with a program where they said that they want to study the toxicity of new and emerging tobacco products. Because acrolein is one of the molecules which is generated by tobacco smoke in large quantities. And because of our background in aldehyde metabolism, we were approached if we wanted to partner with American Heart Association to study the toxicity of new and emerging tobacco products. So myself, Arnie, and Dan Conklin, we got involved in this process, and we published a series of studies looking at the contribution of aldehydes in the toxicity of some of these molecules. The acrolein is one of them that we studied. Smokeless tobacco product. Dan studied electronic cigarettes and the aldehyde generated by that. Subsequently, after the first cycle, I moved on to do other things, and Dan Conklin is now leading these tobacco product studies. But we do got a couple of grants funded out of one, which helped to do these, take these to study further. Dan had one out of one, and I got an out of one funded of that. But although I'm not deeply involved with many of the other studies, I'm still looking at the aldehyde generated by electronic cigarettes, how they affect atherogenesis. So this is a study that we're doing in collaboration with Dan Conklin. Exposure to electronic cigarettes increases acrolein formation, and it also increases lesion formation, doesn't affect the lesion cholesterol, and it destabilizes the lesion, as evidenced by activation of MMPs in the lesions and accumulation of the CD3 and CD68 positive inflammatory cells, T cells, and macrophages. So now Marina in our group is leading these studies, looking how, like you're doing the single cell RNA-seq, try to tease out the molecular mechanism and cellular mechanism by which these electronic cigarettes, especially the aldehyde, exacerbate atherogenesis. So the other aspect of these, as I said, we looked at endogenous molecules, regulation by tobacco products, but the core issue is whether the other environmental contaminants affect endothelial activation, which or endothelial injury, which is the first step in the atherogenic process. And if so, can we develop a program where we can look at the biomarkers of vascular injury and atherogenesis and see how that progressed with the exposure to these environmental chemicals. So the reason that we are interested in environmental chemicals and heart disease is because about 9 million people died prematurely worldwide due to environmental pollution, and cardiovascular disease is the leading cause of death. And out of these 9 million people, 6 million deaths are attributed to airborne toxicants. So if you look at the air pollution, you can see that volatile organic compounds, which are essentially, majority of them are either carbonyls or they are metabolized to carbonyls. They are prevalent in the air, both indoor and outside. And among the VOCs, if you look, aldehydes and then petroleum products, BTEX molecules, which I'll talk a little later, constitute about 50%, and 50% of the other ones are PHs. So our initial studies are focused on these BTEX molecules and aldehydes. And these VOCs, the reason we are interested in these are abundant at Superfund sites, and people living near the Superfund sites have increased risk for diabetes and stroke. So that's the reason we are interested in studying the VOC toxicity. Some of the VOCs of interest to our group here in the Superfund program, acrolein and crotonaldehyde, they by themselves are carbonyls. And then the molecules, like, as I said, BTEX molecules, benzene, xylene, ethyl benzene, and so forth, styrene. And then some like the molecules like butadiene, which is used in the rubber industry, can be metabolized to acrolein. And vinyl chloride and TCE are chlorinated VOCs, but their metabolic products are also aldehydes. So we got funded by NIH to study the VOC toxicity, especially the cardiovascular disease and metabolic disorders in 2017, and then we got renewed in 2022. So the structure of this program is that we have two biomedical projects where we study the population-based studies and more mechanistic lab studies. And then there are two environmental projects which study the levels of these VOCs in the environment, developing new devices to do that, and B, how to mitigate these VOCs from the environment. And then complementary to that, we have multiple cores, which community engagement core, which really helps us do the population-based studies. And more importantly is the research translation, research experience and training coordination core, which is led by Dr. Hine and Dan Conklin, where we work very closely with the T32 program. And essentially we are looking, training the young scientists to study the cardiometabolic toxicity of environmental chemicals. So I'll share some of the data generated through this program recently, and they are especially for the biomedical project side. Essentially, we have enrolled in the first phase 730 participants, and we'll be extending this study to 1,200 participants. And our project one is population-based studies, where we are prospectively going to study these people over a period of time in aim one, and aim two in A1, and then we will also measure. So we will have, I will show you some of the data. We will have various physiological and biochemical measurements of the cardiovascular and metabolic functions, as well as the individual assessment to exposure, which we measure by measuring VOC metabolites in the urine. At the community level exposure, we are doing the wastewater monitoring, looking at the VOC and VOC metabolite in the wastewater, and looking at the disease outcome, mortality, and hospital admission, and correlate them with the VOC exposure. And then, of course, then we will have the longitudinal follow-up in time. And at each visit, we get the demographic information, we do the physical exam, we do a lot of clinical chemistry measurements, and then we measure biomarkers of inflammation, endothelial toxicity, hepatotoxicity, and so forth, and thrombosis. And we see whether, because VOCs are generated in large quantities, whether these processes are, what is the contribution of tobacco exposure to these markers of disease. And complementary to that, we have animal studies where mice are exposed to individual VOCs to test the plausibility that a given VOC is sufficient to cause the changes seen in the human studies, and then tease out the cellular and molecular mechanisms. And our studies primarily right now, the one I'm going to share today, are focused on the endothelial toxicity, because apart from being critical for atherogenesis, endothelium also serves as a barrier to separate the blood from circulation to going inside the organs, controls the blood pressure, controls the regulation of inflammation, and is critical for angiogenesis and vascular repair. So one of our recent studies showed that exposure to VOCs increases systolic blood pressure. These studies were led by Captain McGraw, who has now gone to Columbia to do her postdoc. And what she found was acrolein, urinary metabolite of acrolein, this 3-HPMA, is associated with increased systolic blood pressure. And then metabolites of acrolein and butadiene are associated with the impaired vascular function, RHI, which is an index of endothelial dysfunction. And complementary to these studies, Marina Malovichka in our group is leading the studies looking at the microparticles coming from either the endothelial cells, platelet, and or they can also be coming from the immune cells. How do they contribute to the vascular toxicity? So in Caitlin's study, it was dysfunction. Here, she's studying whether it's a vascular injury. And so for this specific study, we analyzed 667 total participants. And then we especially look at the non-smokers because we wanted to dissociate the effect of smoking. And we found actually in the non-smokers, the associations for many VOCs were much stronger than what we were seeing in the smokers. And in the lower panel, you can say endothelial microparticles and activated endothelial microparticles, they are increased, especially in the age population. So the aged people are much more susceptible to VOC-induced endothelial injury. And especially looking at a class of compounds, we looked at the BTEX molecules, and we found that several of these endothelial microparticles were associated with the metabolites of benzene, toluene, thylbenzene, and xylene. So endothelial microparticles, activated endothelial microparticles, lung endothelial microparticles, and activated lung endothelial microparticles. And to test the plausibility that, again, before I get into that, We also looked at, stratified the data by sex, and we found that the toxicity was associated with both males and females, but especially for endothelial microparticles, it was much more pronounced in female. We don't know why. So then we can say, like, okay, can we recapitulate this with the individual benzene or xylene exposure? And indeed, we found this similar to humans, exposure to mice to these individual chemicals, in this case, benzene and here xylene, increase the circulating microparticles, suggesting that exposure to these molecules causes endothelial injury. And same thing we could do at the cellular level. We looked at the apoptosis of endothelial cells, and that was associated with when we incubated the cells with one of the benzene metabolite, muconaldehyde, and we found that there was increased microparticles associated with muconaldehyde. And some of these studies are being led by Samantha, who's a graduate student in the pharmacology program. So when you have endothelial injury that cannot be sustained for a long period of time, there has to be a mechanism for endothelial repair. And so the question we asked, whether exposure to VOCs could also compromise endothelial repair. And what we found that when you have the VOC exposure, these VOCs get metabolized, that causes endothelial injury. These metabolites can also mobilize the cells from bone marrow, like endothelial progenitor cells or circulating angiogenesis cells, the same thing. And then these could be involved in the endothelial repair. So we look for the markers of endothelial repair in that context. And these studies are led by Dan Griggs, who just graduated from our public health program and has now been recruited as faculty in Department of Medicine. So again, we recruited these participants from our Superpharm studies, and what we found that exposure to VOCs increased the circulating angiogenic cells, again, in the whole population, as well as in non-smokers, and give you some, especially some of the chemicals which were associated with changes in the circulating angiogenic cells, were benzene molecules, metabolite muconaldehyde, acrylonitrile metabolite CYMA, and ethyl benzene metabolite PGA. And similarly, if we expose the mice to just benzene, we can still see similar to humans, this decrease in the circulating angiogenic cells, suggesting that exposure to individual, at least this chemical, and same thing we found with acronym, is sufficient to deplete circulating EPCs. So the question was, okay, we are seeing a lot of these toxicity with these VOCs. Why don't we pick up one molecule and look in the experimental animal conditions in the disease process? Because in the human studies, we were studying only the biomarkers of study. So we did 3 different studies. One, it was exposure to VOCs. We studied insulin resistance. And this study were led by Wes, who since moved on to Germany to do his postdoc and now has been recruited as a faculty over there. What he found that exposure to benzene causes impairment of insulin resistance. He found both GTT and ITT were perturbed by benzene exposure, and this was mediated by oxidative stress. And Tim O'Toole was the lead person on this project, so he was dealing with this project. In my group, we collaborated with Steve Jones to look at the pressure overload induced heart failure model, and Igor Zelko led these studies. And what we found that exposure to benzene decreased the ejection fraction in these mice subjected to pressure overload. Then another study that we are looking at the effect of benzene exposure on atherogenesis, that is led by again Igor Zelko and now subsequently Brandon Taylor is looking at the immune component of these lesions in the benzene exposed mice. So I give you a little insight just in the next couple of slides before I wrap it up to say how exposure to benzene changes not only the lesion size, but lesion inflammation and stability. So we did the single cell RNA-seq of benzene exposed mice. And what we found that there were 23 different populations of cells we could detect, several population of T cells, endothelial cells, and macrophages. And what was really striking was that the T cells were increased in the benzene exposed mice. So that we got us interested in looking in some of the greater details. And then Igor started analyzing some of the data. And here is the group data. So T cells and smooth muscle cells were the cells which were mostly affected by benzene exposure. And then we looked at this genes associated, especially some of the genes associated with inflammation with T cells. And as you can see, in some cases, there were profound induction of genes in T cells, whereas it's in the other cases, genes were completely knocked down by the exposure to benzene. So it's a very complex mixture of things which is going on right now. And we're trying to design some of the cellular studies to get a better handle on the molecular mechanisms. Then, because we did the single cell RNA-seq, we could also look at the interaction of these T cells with the endothelial cells in the plaque. And then Igor did this analysis, which is called cell chat, where you're looking at the communication of the signals from endothelial cells to the immune cells. And then he could identify several targets which could be facilitating these processes. And especially one of the things that we narrowed down is IL-7 receptor. So we think that it is a critical molecules in changing the inflammation in the lesion by benzene exposure. And then we're planning a series of in vitro studies to tease out the mechanisms. To summarize, when we have the carbonyl exposure, it can cause insulin resistance, like I showed for benzene, and then it can lead to secondary diabetic complications, which can lead to endothelial injury. I didn't share much of the data on platelet, but they also cause platelet activation. And then exposure to carbonyls causes protein misfolding, which leads to ER stress and subsequently endothelial activation. And then endothelial injury could also activate the endothelium, and so can the platelet activation. And this leads to atherogenesis, which subsequently leads to ischemic heart disease and heart failure. And I did not show the data today, but another series of experiments that we are doing is looking at the heat shock proteins as an adaptive stress response to mitigate the effects of the VOC-induced endothelial dysfunction. So with that, I wrap it up, and I want to acknowledge the funding from NIH. And this is a little dated, but when the NIH Superfund Program Officer visited us, I think just before COVID, but many, many people contributed to this study. I could not acknowledge everybody individually, but Nalini over here does a lot of our cellular studies. She's the backbone of our cellular studies. Millie does a lot, apart from administrative duties, she processes all the atherosclerotic lesions. And David Whitaker and Paul's team do a lot of studies looking at the mass spec study, looking at the biomarkers of exposure. With that, I wrap it up, and I will be happy to take questions that you have.
study. Welcome any questions.
Yes,
I have a couple of questions.
sure.
So for the VOC population they study, you are using urinary metabolites as a marker of exposure, is that correct?
Correct.
So how do you know that these people are more exposed because they have more VOC in their urine or they're just better excretors?
You are absolutely right, because if there are genetic alterations, like they are more metabolically active, we don't know. Because we are the polymorphism in some of these VOC metabolizing enzyme is not very prevalent, except for some of the aldehyde dehydrogenase ones, which are there in mostly in China. So we don't think that is a major contributor, but in principle it could be. We cannot dissociate that part.
What about transporters?
We don't know. You're absolutely right. We don't know the transport part, right? So we are also looking at the cumulative exposure over a period of time. We are looking, we have not done that, but in the next phase we are going to. We have collected toenails and hair to look at the cumulative exposure. We will also look at the cross-linking of these proteins in the tissues and the cells.
Yeah, that's always my problem, like how does urine give?
Yeah, right. So, but I mean, like you have to start somewhere, right? Yeah, yeah.
The second question was for the other study you're proposing to look at the effect of VOC, I believe, on I think air pollution. You are you're probably getting most of your subjects from the Kentucky area.
Everything, yeah, all these are from Jefferson County.
Right, and we know that there is quite a bit of metal exposure there, which you have shown that also gives rise to many of these outcomes. How are you planning to get rid of the effect of metal exposure?
Yes, so we wanted to study in addition to VOCs, we wanted to study metals. And we proposed that in our Superfund application in 2013, and we got hammered on that. They said you want to do target study, you don't want to do everything in one go. Now, the whole dynamics has changed since then. Now it's like, okay, you should be doing exposomics, right? So now don't study just only one class of compound, just you look in totality. Yes. So now increasing from VOCs, where now we are doing mercapturomics, where we are looking at mercapturic acids instead of looking at 12 VOCs, right now we have gone up to 500 metabolites coming from like 70 different VOCs. Not VOCs, but mercapturic acid driven molecules. So we are expanding that. We do plan to look at the metals because when we want to actually do the whole exposomics, so when we have more resources, we have biobank samples, and we do want to look. Actually, I talked to him, Lukai, sometime back. We have not been able to do that, but yes, we do want to look at the metals, and we want to see the association because some of the metals could also be associated with some of the vascular injury or markers of endothelial activation of vascular injury. So we don't rule it out. We definitely think that we should study.
Thank you. Amazing.
Yeah, it's okay. I want to follow that.
Yes.
Question for the metal issue. Actually, it's not necessary to the individual have to export to some heavy metal. And even the VOC project change the signaling pathway, also alter stress that make the essential metal like zinc, copper, iron, better like the balance, distributes the balance. That also is it's very important. Yeah.
And absolutely, and metals also metals also catalyze lipid peroxidation, right?
Yeah.
So the Fenton reaction is iron catalyzed and or copper catalyzed. So most of the lipid peroxidation, you need a catalyst like.
Not indirectly, in fact.
Yes.
So if you have the sample, we really need this thing, what measure.
We definitely have the samples. We are trying to get resources to do that because not only I just showed you data of our study. We are now collaborating with Mesa. We might be getting samples in the next couple of weeks. So that's this atherosclerosis study looking at air pollution. And they have the metals data for 6,000 people. And they have atherosclerosis readout and vascular and biomarkers and so everything. Inflammation for last 20 plus years, 4 visits. They don't have the VOC data. So that's what we are measuring. And that's really going to open up a lot of new channels, how to VOC by themselves or in conjunction with metals, how do they contribute to atherogenic process. We're also working with regards, which is a core of 32,000 people looking at stroke. And we have a conversation with them. Probably by the summer, we will get samples from them and essentially looking at the association of VOCs and metals. They have metals in their studies, at least a subset of that. And then we want to study the VOCs and see how this VOC metal interaction happens. If NIH did not ding us on that, we would have done a lot more. So they actually slowed us down. Any questions online?
There are no questions online.
Well, if not, thank you.